Hello and welcome to another video on plant physiology by Perfect's course. This is Preetinder Kaur, and so far what we've done under this topic is we've done what is the process of transpiration and how does the structure of xylem help in that. So we're going to start on with that, and we already did a model of the potometer which is used to measure the rate of transpiration. So what we are going to do first in the video is going to be an experiment which is known as the Renner Experiment. So what happened in this experiment was exactly the same apparatus was used as in a potometer, and this experiment was conducted by Otto Renner in 1912. But there were a few changes. The first change was that there was a woody chute that was placed in the potometer. After a while, there was another change that was done. That means a clamp was put on the chute to control the upwards flow of water. To control the upwards flow of water. Then after a while, another change was introduced, and that was the top, which is having the leaves, the top of the shoot with the leaves was cut and removed. And finally, another change was done. Instead of the leaves, which were not there anymore, a vacuum pump was attached to the shoot. After a while, Otto Renner recorded the change in the rates of transpiration, and this is what he found. So on x-axis, if you put number of hours, you put 1, 2, 3, 4. And on the y-axis, let's take them 10 and 20. These are the rates of transpiration in centimeter cube per hour. So he observed that initially the rates were going fine, the transpiration was reducing. But as soon as step number 2 was done, that means the clamp was put on the chute to control the upwards flow of water, the rate of transpiration dropped. After that, it started to increase again, but in this direction, and this is where the third step was implemented. That means the top with the leaves was removed. So naturally what should happen is the rate of transpiration should come down, and that is exactly what was happening. And at this point, the fourth step was implemented. The vacuum pump was attached, so the rate of transpiration increased and became stable after a while. So you can see that leaves have a very important role to play in transpiration, and that is what this experiment shows. So the next thing that we need to do is learn how tension in leaf walls is useful in the transport of water. So there are a few steps that you need to know. The first thing is that on the surface of the leaf, the water evaporates. And this usually happens during day. Now when the water has evaporated, there will be forces of adhesion that will come into play. And adhesion is going to draw the water from the nearest source possible to the leaf. And what is the nearest source of water to a leaf? The veins in it. So the adhesion is going to draw the water from the veins. And what is there inside the veins? Xylem is there inside the veins. So what do the xylem vessels inside the veins actually do? They suck up water even under low pressure. So the xylem vessels in the vein, they suck water in the upward direction even when pressure is low. And what will this lead to? Even further reduction in pressure. Further reduction in pressure. Due to this low pressure, this extremely low pressure, it generates a pulling force. And this pulling force goes from the leaf to the stem, from the stem to the root. And the name of this pulling force, let's call it transpiration pull. This is the name of that pulling force, which is there because of further reduction in pressure. So we can say that water transport via the xylem, it strongly depends on adhesion and even on cohesion. So this is about water, but what happens to minerals? Now what happens to minerals? You know that water in the roots, it is absorbed by osmosis. So let me write it here. Water is absorbed into the roots by osmosis. That means that the concentration of water in the soil is more than the concentration of water in the root. So naturally, water tries to go from an area of high concentration, which is soil, to an area of low concentration, that is water. That is the process of osmosis. But what about minerals? Now, the minerals are present in a higher concentration inside the root as compared to the concentration in the soil. As a result, when minerals have to move into the root against the concentration gradient, they use a new kind of transport. So that is against the concentration gradient, and that kind of transport is known as an active transport. For active transport, you need something that can help you transport those minerals against the concentration gradient from the soil into the root. And for this purpose, you have protein pumps that are present in the plasma membrane of the cells. So the special protein pumps, they help in exchange of the mineral ions. And for each mineral ion, there is a special pump. Now sometimes soil particles may hold on to certain ions very strongly. So those minerals may not move so freely from the soil into the root. So what happens is they move from the soil into a special kind of fungus, into the hair-like structures or the hyphae of a fungus, and from the fungus then they move into the root. So basically the fungus is helping to transport the ions from the soil to the root, and in exchange the root gives the fungus lots of sugars and other materials. So this positive mutually beneficial relationship between the fungus and the root is known as a mutualistic relationship. So this is how basically the relationship is there between fungus and root, and this is how in short the minerals are exchanged. So what we're going to do in the next video is we will discuss about this entire process of water and minerals, what happens at the root end and what happens throughout the xylem. So we'll be discussing that in detail. So keep watching these videos, and that is all for this video. Don't forget to visit us at perfect-scores.com and share and like our Facebook page. And for any suggestions or feedback, you can mail us at Gmail. Thank you so much for watching this video.